Good morning, ladies and gentlemen, boys and girls. Today, we are going to talk about a little bit more detail about the stages of the cell cycle. All right. So, why don't you guys go ahead and get out your handy dandy notebooks, and we're going to talk a little bit about the different stages of the cell cycle. All right. So, I'm actually going to give you a moment right now. If you're just getting out your notebook and this is the first time seeing this video, give you a moment to go ahead and copy down what I have already. All right. Mostly just this big circle that we have divided here with little arrows and boxes and stuff like that, just so that you can be paying attention and listening to me as we're talking about it. The beauty of this video is that you can pause this video, write that stuff down. I'm going to let you do that right now. Pause the video. All right. So hopefully you are done with that. And what we're going to talk about now are the different stages. All right. So first of all, let's go back to the big picture. Why does the cell need to do this? What is happening here? All right. One of the reasons, or the whole idea of this big picture, the purpose of the cell cycle are the stages, the checkpoints, the things that need to happen for any cell in your body or any cell that exists to go from one cell to two cells. And there's multiple reasons why that might need to have happen, depending on the organism. Some organisms, there's basically three major reasons why. Number one, why a cell needs to divide. Number one is to grow. All right, you are not as big as you once were. It's not because your cells are getting bigger. You are just gaining more cells as you get older. All right, as you grow, your number of cells are increasing faster than the rate that your cells are dying, because cells do have a short lifespan. All right, so number one would be growth and development. Number two would be to replace your old and dying cells. All right, not only do you need to grow and get bigger, all right, you are constantly losing cells, depending what type of cells. Some cells last only a few hours. Some cells can last many, many years. All right, it just depends on what happens. And if you need to replace those cells, then we have to go through, the cell needs to go through the stages and the checkpoints and the things it needs to do to go through the cell cycle. Now, I should point out, as I'll talk about later, there are some cells that don't go through the cell cycle, like nerve cells and brain cells do not, once they are gone through the cell cycle up to about your birth, they do not go through the cell cycle anymore, which we'll talk about that in this video. All right. And the third reason why an organism might need to go through the cell cycle is to reproduce. If you are a single-celled prokaryotic bacteria, those very, very simple cells that have no nucleus, don't have any extra organelles, right? They completely divide using just the cell cycle and mitosis, which is a big key word we'll be talking about in a little bit. All right. So that's the three reasons why a cell might divide. All right. To grow and develop, to replace an old cell or damaged cell, and to reproduce if you do asexual reproduction. If you do asexual reproduction, then you are going where the offspring exactly like the parent, then you are going to go through this cell cycle in this manner that we're going to explain today. All right. So the big goal for us today is to talk about how we're going to go from one cell into two cells. I get a cut on my arm. I need it to heal. All right. I lost a lot of cells during that gash. All right. I need to regain those cells to fill in that space. I need the cells to, those cells near that cut to go through the cell cycle, to gain more cells. I need to divide one parent cell into two identical daughter cells. Now, when I say identical, I mean identical in size. I mean identical in number of mitochondria. And most importantly, identical in their DNA. Every cell in my body has the exact same copy, or at least should have the same exact copy that makes up John Richardson. All right. Every cell that needs to be formed needs to have that same code. All right. So let's talk about the stages through this as we go through this cell cycle. I'm going to start kind of from the top here. We're going to work our way around as we go around the cell cycle, talking about the stages. And we're just going to highlight some of the main things. Questions you need to think about, or questions I might ask later, is obviously what happens in each stage and why is it important for the cell? And one way to think about why is it important is what would happen if there was a mistake. So one we'll talk about is S phase. Well, what would be the problem if there was no S phase? Why would that be a problem for the cell? And that's what we're going to discuss here today. All right, so let's go ahead and get started with our very first stage, probably the biggest part of the pie, all right, and that is gap one. Now, I should point out, notice how these are different sizes, all right? They're all different sizes and it's all going to look different basically in every diagram, but it's based on time, all right? And every cell's a little bit different, so this is kind of an average, all right, about how long it takes for this cell cycle. This whole cycle for some cells takes minutes. Some cells, it can take a lot longer than that. All right, but it's spending the biggest chunk of its time in this very first stage here, what we call the gap one phase, or just G1. All right, so we're starting with one cell here, and we're going to start in the gap one phase. And like I said, we're going to think about what happens, just the big picture. All right, there's a lot of other stuff that's happening. There's a lot of other stuff that's happening. We're just going to hit some basic stuff as we go through it. All right. So one of the things that's going to be happening in G1, and notice it's taking a lot of this time, all right, is it's going to grow in size. All right. Which makes sense. All right. If I just take a cell and divide it in half, or if I have this big cell and I divide it in half, now I have two smaller cells. And if I take this smaller cell and I divide that in half, I'm going to have even smaller cells. All right. So we needed to kind of swell up just a little, just swell up so that when we split it in half, it looks identical to the original cell. Not just in the DNA, not just in the organelles, but also in its size. So it needs to swell in size to kind of make space, but it's also making a lot of extra space, for one of the things I just said, to make copies of its organelles. Organelles including the Golgi body, the endoplasmic reticulum, the mitochondria. If that cell had 15 mitochondria, both daughter cells need to have 15 mitochondria. We want identical cells, all right? So we are also going to copy the organelles and cytoplasm. That's a lot of stuff to do. I have to make two of everything. All right. If I'm going to have two of everything, all right, so each of the two cells can have one nucleus. The other one can have a nucleus. The other one can have this. This one can have. They both have that be identical to each other and to the original parent cell. All right. So that's gap one. We swelled up in size. And we're getting bigger. All right. That lets us move on to the next stage here. This is a very crucial stage, especially. This is where a lot of mistakes happen. So as we go through this here, we're going through G1. All right. We're going through G1 to make sure everything's going okay. All right. This is super important for your cells to be able to go through this process. There are checkpoints. There are little proteins and enzymes that our job is going to basically make sure, like, hey, did those organelles replicate properly? Are we a big enough size right now? Are we doing everything? Is everything up to date? Are we on task? All right. And so these checkpoints at the end of kind of each stage within that cell, like, okay, all the boxes have been checked. We are now ready to move on kind of the next set of checkpoints, all right, our next set of to-do list for the cell to be able to divide, all right? And that next place that the cell is going to kind of next set of checkpoints or boxes to check is what we call the synthesis stage. Or simply S phase. All right, so now we are in S phase here. All right, during S phase, this is a crucial part here. All right, during S phase, we have DNA replication. Now, we've talked about this in class where we have DNA helicase unwinds the DNA polymerase comes in and adds those free-floating nucleotides where we start with one parental strand of DNA to create two identical daughter strands. All right. So we have here what we have is DNA replication. If you don't remember those details, I highly recommend pausing the video right now and taking 10 seconds, look back at your notebook and just quickly look at it. Just pause it, just refresh yourself on what that means, what some of that stuff is. So I'm even going to pause for a second just to give you a chance to pause the video to look back in your notes for a second. Okay, so if you had the opportunity to look at that, DNA replication is going to happen here. All right, so this is what we're going to have. We're going to have our DNA. So here's my DNA, right? DNA or helicase, DNA polymerase comes in. This would be in its chromatin form, right? And then what's going to happen after replication, we're going to end up with 2 identical strands of DNA. All right. If you remember that those identical strands are held together by that paper clip. That paper clip there is called the centromere. All right. And now we have our sister chromatids. We have our sister chromatids. All right. So we went from not chromatids to sister chromatids because these are identical strands to each other. Why do we need to do this? Why do we need 2 copies? Because we're going from one cell to 2 cells. And each cell needs to have a same size, same number of organelles, and same DNA as each other and the parental strand. All right. So we've gone through S phase. We've gone through DNA replication. There's a lot of proteins that check that work. Did everything go okay? Maybe DNA exonuclease comes in and fixes a few things. All right. Once it's kind of all good, we've checked all the boxes. We have our checkpoint done. We are ready to move on to G2. Notice this is another big chunk of the pie. This is what we call the gap 2 phase. All right. And the gap 2 phase is going to be very similar to gap 1. We are going to continue to grow in size. We are continuing to do a lot of other things. Now, there's a lot of other things that happen in G2, but the 2 high things we're going to highlight is that it grows in size and a new structure forms that's going to be very important when we go into the details of this last phase. All right. Something called the spindle fibers. They are formed kind of at the end of G2, kind of at this last little part of G2. We have the spindle fibers form, right? We have these spindle fibers form, and it creates, or it's kind of going through this phase as it grows in size. It's all about this here. All right, I'm going to pause real quick here when I'm talking about this because I want to highlight something here. Everything we've done up to this point has been in preparation for this one here. So let's say we went through G1, finished that off, had a new set of checkpoints, checked that off, went through G2, checked off everything we do. We're ready to go into this last phase. Everything we've done so far has been in preparation for this very last stage. Notice we are still 1 cell. We have 2 copies of DNA. We have extra organelles. We have extra cytoplasm. We're really huge at this point, but we still are only 1 cell. We haven't actually done the dividing yet. All right. Everything has been in preparation. All of these stages right here, all this prep work is called interphase. Interphase is made up of G1, S, and G2. It is all the prep work that the cell has to do to get ready for this final phase of the cell cycle. All right. And so now we're ready to go into this final phase here. This final phase is called M phase. All right. It's called M phase. Or in this case, for us today, it's going to be what we call mitosis. All right. Mitosis is, now we're ready to divide. We've done all the prep work. We packed all the bags. We cleaned the house. We did all the, we turned off all the lights. And now we're ready to do the darn thing. All right. We prepped everything we needed to do. All right. And now we're ready to do cell division. We will spend an entire day in class talking about the very specific steps and stages within this stage, right? Prophase, metaphase, anaphase, and telophase on another day. All right. All we need to know right now is this is the actual dividing. We have now taken one cell and we divided it into two identical cells. We actually did the cell division. All right. So now we have gone through the cycle. Now, even after we've divided, there are still check boxes that we have to make sure. All right. Where there are still boxes that we need to check. All right. And make sure everything's done. If every check's been checked or every box has been checked, cell cycle has been complete success. All right. And we'll keep going. If I get a cut on my arm, I'm going to need a lot of new cells. Each one of those new 2 daughter cells will become the parent cell, and we'll do this again. We'll go from 2 to 4 cells, and then from 4 to 8 cells. All right, and we just keep going. But eventually, those cells need to stop. I can't divide or stop, or I can't keep dividing forever. Eventually, that cut heals. I fill in that space with cells, and it's healed. And I need the cells to stop and start doing their function. Because cells have a hard time. They can do a little bit of their job, their primary function. Like a liver cell does liver cell jobs, kidney cells does kidney cell jobs, heart cells do heart cell jobs. All right. They can do them a little bit during this G phase, but they're kind of multitasking. I'm doing my job and I'm also dividing. All right. It's a little bit hard for it. So now it needs to stop dividing. All right. Well, this cycle it's a cycle. It's a circle. The circle has no end. All right. And so what it has to do is it has to jump out of the circle. All right. And what it will do is it will jump out of the circle sometimes, out of the phase here, typically out of actually one of the G phases. I'm just going to draw it over here until we get to what we call G0. All right. Now G0, or what we stand for, is gap 0. All right. There is no division. There's no cell division at this part right here. At gap 0, there is no cell division. We stop dividing. All right. And the reason why it stops dividing is because it doesn't want to multitask anymore. It wants to go back to its primary function. A skin cell doing skin cell functions, a muscle cell retracting and relaxing, all right, doing the job it's supposed to do. A neuron cells jump into G0. Their job is to produce neurotransmitter hormones, right? Most cells are going to jump out of G0. Some cells, like brain cells and neuron cells, just stay. Once they jump out, they stay in G0 forever. All right. At the same time, like my stomach cells, right, they need to replicate really often because the stomach acid needs me to replace it. And so what happens is my stomach cells will be in G0, right, doing their function. So it performs cell functions. It's performing its cell functions, its jobs. And then what it will do is like, oh, okay, we're running out of cells here. I get a signal that I need to jump back in. It will jump from G0 back into the cell cycle and get that process again. All right. So sometimes if the cell wants to stop, it jumps out and it will jump back in if it needs to go through the cycle. And when it's done kind of dividing, or what it needs to do, it will jump back out, stop dividing, and do its job. All right. It has a hard time doing both. Again, the cell can't really multitask. It's either dividing or doing its job the best it can. And so it will either jump out of the cycle into G0, do its job, or into the cell cycle, go through G1, S, G2, M, make sure to check all those boxes. All right. That was a long video. I understand that. That was a long video, but there's a lot of things happening here. Remember, the whole purpose of this is to go the steps, the boxes to check for one cell to become two cells. Not just two cells, two identical cells, identical to each other and to the original parent cell. Identical to each other and identical to the original parent cell. And it's going to go through interphase and mitosis. Remember, interphase, which is G1, S, and G2, is all about prep work. Growing in size, copying organelles, copying DNA, growing spindle fibers so that we can actually do mitosis. And we'll talk about that actual division of going from one cell. So it's all about prep work, prep work, prep work, divide the cells. And if it stays in the cell cycle, then each new cell will go through prep work, prep work, prep work, divide again. Or it will jump out into G0 and perform its normal duties. All right, that was a lot. If there is something on this board, like I don't understand what he was talking about with sister chromatids, please make sure that's the beauty of your video. Go back and watch it again. All right. Otherwise, you can ask me some questions in person. Go back and watch the video if you need to. All right. Thank you very much, and good luck, and may the science be with you.